Okay, so good morning everyone. So I'm presenting today my preliminary result from a master's studies I initiated earlier this year. And I've been working on the population genetic structure of the common smooth-hound along the South African coast. So just to start off is that the common smooth-hounds, the consequence of them being much more on threat is that they are much more valuable and have higher preference over other species by consumers. They are also caught just bycatch in the headlong line as well as trolls, as well as an increase in demand, as well as an increase in exploitation for shark fillets in Asian and Australian countries has also been the contributing factor. And these attributes have in turn resulted in the species being listed globally as vulnerable by the International Agreement for Conservation of Nature, REDS. So recent reports suggesting wild population declines among population stocks of commercial shark species due to legal as well as illegal fisheries has resulted in us hoping to actually try and understand the levels of genetic diversity as well as the patterns of gene flow, so we can be able to discern future management units for conservation and as well as for management. And currently, the population genetic structure for the common smooth-hound shark, both globally and regionally here, particularly in South Africa, is not known. So that is why we are set out where the aim of our MARCH project is, among the aims, is to actually test for gene flow being the reproductive, which is the movement of individuals from different populations into other populations, and then they mix and they mate. And that in a whole creates a one larger and limited population, which is a hypothesis of Bayesian, or the alternative hypothesis that it is an existence of two or more stocks, indicating that there is some degree of population subdivision along the South African coast. And to achieve this, what we set out to do was to genotype the 7 sampling site that we managed to get our hands on currently, of a total of 110 individuals, and to assess the levels of genetic diversity amongst the study populations. And the ultimate objective being that we would like to evaluate the degree of population differentiation between the populations along the South African coast. So the 7 sampling sites that we have, although the sample size are fairly small, is that we have samples from Nangabonde populations as well as Robben Island, and these two populations are actually above the traditionally recognized biogeographical barrier, which has held strong in restricting gene flow for many marine organisms. But interestingly enough is that this barrier did not seem to restrict any gene flow for the toe shark as well as the copper sharks, since samples from around about there did not show any population differentiation from samples from up to Namibia. And below that barrier we have samples from False Bay and Galway, and then from Garlis Bank. And these 5 populations over here represent the Atlantic Ocean populations. And we also have samples, we had the Jeffreys Bay population, which was going to represent the Indian Ocean populations. And we managed to get samples from the Sharks Board for a different project, and I've decided just to add them on for this particular, for the presentation, just to illustrate the differences that we can see with the Atlantic populations versus the Indian Ocean. So, as the species has very limited genetic information, is that in order to conduct a population genetic study, is that we had to first develop molecular genetic markers, which are a prerequisite for population genetic study. And we decided to go through the cross-species amplification route, which is a transfer of primers from closely related species to the target species, the one that you're trying to actually develop markers for. And microsatellite markers, which are a repetitive sequence of 1 to 6 base pairs in the genome, and they're highly variable, which obviously that's why they're suited for population genetic study. And from these 2 papers, luckily in 2012, markers that were published for the Mustelus henlei as well as Mustelus canis that were actually showed to be closely related to the Mustelus mustelus. And we selected those markers and we did cross-species amplification. And then here I'm just showing an example of the MH2 from the Mustelus henlei marker, as well as the MH9, which showed successful amplification on your agarose gel, which is a gel that can discriminate between fragments that are around about 100 to 150 base pairs. And markers such as these that show robust amplification on the page, on the agarose gel, would then have to be evaluated for polymorphism to see if these markers actually demonstrate alternative forms, which we refer to them, of different band sizes, which we refer to them as alleles. And in here now, if you take image 2 and you run it, we use a higher resolution gel, such as page gel, which can discriminate between fragments that are at least 1 base pair difference. And you can see that this marker is polymorphic, since you can distinguish around about the number of different fragments, different base pairs there. And then after evaluating for polymorphism, we had to select markers that were polymorphic to actually establish two multiplex systems, which constitute of 6 microsatellite markers, each of which we then use predictable electrophoresis to genotype the study populations, and then we use a programmatic scanner to do. And then there is just an example where you can see that this is a virtuocyte, there might be an individual that represent two different alleles of different fragment sizes. And so just to look at the genetic data analysis is that we had to address, we first had to address the question of whether there is one stock, if there's two or more stocks. And then to do that, we had to test the hypothesis of one stock, and over the alternative hypothesis that there's two or more stocks present along the South African coast. And then we conducted the molecular genetic data analysis, and the statistical significant level was at a 5% nominal level. And then yet again, you have to address whether the statistics are significant or non-significant, and should they be, it means yes, there is population structure, and therefore you can say with certainty that there seems to be, they can be managed as a separate unit. However, should the statistic not be non-significant, it means that you just have one large accumulating population. And obviously some have said that you can manage a population as one unit. However, that's debatable, since that genetic connectivity does not necessarily imply that your populations are also demographically dependent. And both of these results obviously has implications for the management and trade monitoring of species. So looking at the results for the genetic diversities, what we detect currently, although the sample size, as I mentioned, is very small, is that we've managed to detect comparable levels of genetic diversity across all the study populations. And in here I'm just representing them as in the form of allele patterns, where we use the genetic indices such as the number of alleles, which is the average number now. We've taken all the multi-locus genetic data, and you can see here that the mean number of alleles are actually comparable with regards, and then the Rouvay island population seems to have the highest number of alleles. The number of effective alleles here, which are the alleles that can be present at that population, obviously it seems to be obviously comparable across populations. And the Shannon's information index, which quantifies genetic diversity in one population relative to the other one, also seems to show that it seems to be also comparable, in telling us that obviously we suppose that the genetic diversity indices are actually comparable. We're just looking at it. And then the number of private alleles, which are alleles that are unique to a particular population, or only segregate in that population. And you can see this is they're very comparable, with an exception of the Robben Island population that seems to only have the highest number of those alleles that are segregated in that population. And then expected heterozygosity levels, which is also a measure of polymorphism within the populations, as well shown to be consistent, shown to be comparable across generation. And then the dip that we see here in all of the genetic diversity indices is a result of the small sample size that we have. So looking at the results of population structure is that we use the analysis of molecular variance, which is a technique of which is a method of studying a molecular variation and how it is partitioned amongst populations. And this particular statistical test detected that there were significant differences amongst the populations, which is that moderate 6%, and then also detected significant differences among individuals within populations, which is at 5%. And the largest molecular variance was within populations themselves, indicating that the levels of inbreeding meaning that is the mating of related individuals, was very negligible, or was very low to negligible. So another method to actually assess population genetic structure is that we use a pairwise FST estimate. And now this method requires that you have to predefine your populations, just like the AMOVA, and then it just does it compares a population pairwise to actually determine the genetic distance between the populations. And it also supports the results of molecular variance, as it detected significant moderate differentiation between the Cape population and the Lama population, as well as the Rouvay Island versus the Cape population. Now, interestingly here is that the Stellenbosch population, which represents the Atlantic Ocean, seems to have shown a large genetic distance or large genetic population differentiation in comparison to the rest of the other sampling populations along the South African coast. So now looking at that visually on representing the molecular variation on a 3D vectorial plane, is that this particular the FCA, the factorial correspondence support, now visually shows the patterns of population genetic structure, where now we can clearly see that the different population classes separately from the rest of the populations, which obviously are the Atlantic class, the Atlantic Ocean populations, and that is an Atlantic, and then this is an Indian Ocean population. But interestingly enough, it was that the Jeffreys Bay population, which is meant to be from the Indian Ocean, actually clustered with the individuals of the Atlantic Ocean, and then that just made me that I just need to increase my sample size and use more markers and then just to actually split this particular class, because the factor 1 explains most of the molecular variance. That's why we separate them like based on that into two classes. However, you can go again and separate that one into two other classes based on that plane of that factor 2, which explains most of the molecular variance, where you see that your Lama-Wanda population and Robben Island populations seem to cluster differently from the rest of the population that have Bay, Jeffreys Bay, as well as Force Bay, which obviously might lead towards the hypothesis of the being weather barrier, which is exchange flow. To additionally assess the population differentiation was that we used the Bayesian clustering analysis. Now with this method is that the program, you assume that it just considers that you only have one population, and then it sifts through your data to try and find the ancestral populations which your current population may have descended from. And then in that, it gives you two colors. Like it will give you a number, what the probable number of populations are, and then we can take it to be them 2. And then we've got the green population as well as the red population. And then we can see here again that the Devon population seems to be very representative of the red population, whereas the other population are a net mixture of both the green and the red, and then the Jeffreys Bay obviously showing similar patterns with the Atlantic population. And this might actually, we still need to actually look at the population in a bit more detail. So just to conclude is that we detected comparable levels of genetic diversity across the study populations, but most importantly that we were able to actually detect a moderate as well as large levels of population differentiation, and as a result we managed to actually identify at least 2 vegetative management units, and therefore consequently our hypothesis of demersal was actually rejected. And since my project also aims to actually characterize microsatellite markers for genetic studies of the Mustelus mustelus, since in the near future we also want to actually study the genetic mating system of the species to decipher of multiple fertility as well as reproductive collaborative. And my project mostly focuses on the Lama-Wanda population, where we will be testing for temporal variation, which is interpopulation genetic changes over time, and we'll be able to compare those estimates with all of the sampling populations along the South African coast. And preliminary observation has it that this Lama-Wanda population is actually phenotypically or morphologically distinct from the rest of the sampling populations along the South African coast, and therefore we also set out to actually test if this population is selectively adapted to its environment, or is it just a mere factor of phenotypic plasticity. Since I cannot say with certainty whether there was a reduction in the genetic diversity estimates, is that I would have to go back after increasing my sample size to actually estimate the historic as well as the contemporary effective population size, since it's the critical parameter in population genetics, since it is where it indicates the rate of loss of genetic diversity, and the effective population size is actually the genetic number of individuals in the population that made to produce the second generation, in contrast to your consensus size, which is just the head count of the individuals. And we'll be also evaluating, and then we'll also be doing population bottleneck, a reduction in genetic diversity as a result of reduction in population size. And then at the end of it all, hopefully using other research outputs, is to be able to formulate recommendations for an integrated conservation management program for the demersal shark species. And with that, I would just like to acknowledge the NRA for funding, but most importantly, our lab and molecular project research group for all their technical assistance and guidance with my project. Thank you.